Hello everyone, a very welcome to Vyas Edification Kota. So today we are going to start with a new chapter of class 11th, that is chapter number 2, Biological Classification. And in this session we are going to discuss each and every question in detail here, which is given in your NCERT textbook. So the chapter name, as we have already discussed, that is Biological Classification. This is a chapter, second chapter from your NCERT, and we have total 12 questions in it, and we are going to discuss each and every question here. So we have first question, very first question of this chapter on the screen, that is discuss how classification system have undergone several changes over a period of time. So first of all, the classification system have undergone several changes over a period of time. First of all, if we talk about the classification system, yes, it has been changed over a period of time, and the classification system for the very first time was introduced by a scientist named as Aristotle. So Aristotle was the first scientist who introduced classification. What he does, he actually classified the plants into three categories such as herbs, shrubs and trees. He also classified animals on the basis of presence or absence of red blood cells. But the system of classification failed because this system of classification given by a scientist Aristotle could not classify all the organisms that were known. So this classification system has been failed. Therefore, a new scientist, another scientist has came named as Linnaeus. He classified the organisms. He classified the organisms into two categories such as kingdom Animalia and kingdom Plantae. So Linnaeus gave us a two kingdom system of classification in which he classified the organisms. He classified the organisms and put the plants into kingdom Plantae and put the animals into kingdom Animalia. This system of classification, the two kingdom system of classification, was also failed because this system of classification could not differentiate between the multicellular and unicellular organisms, also could not differentiate between eukaryotic and prokaryotic organisms. Then finally we have a new system of classification which is also followed now, and a new scientist named as R.H. Whittaker was proposed. He proposed a system. He proposed a 5-kingdom system of classification. He classified all the organisms into 5 categories. He made the classification easy, and he classified the organisms on the basis of some characteristic features such as presence of cell wall, mode of nutrition, habitat, etc. So all these were the characteristic features on which he classified the different kind of organisms into different categories, and he classified all the organisms into 5 categories or big groups, and he named all these 5 categories as kingdom Monera, kingdom Protista, kingdom Fungi, kingdom Plantae and kingdom Animalia. So we have a 5-kingdom system of classification which was proposed or introduced by a scientist named as R.H. Whittaker. So this is how over a period of time several changes has been made into the system of classification. So this is answer, and we are going to write the answer here, that first of all there are several changes has been made in classification system. In classification as we are talking about the classification systems changes, classification system over a period of time, over a period of time, of course. Yes, because at the early time we had only a 2-kingdom classification, then now at present we follow or we have the advanced type of classification, that is a 5-kingdom system of classification. So the first classification system was given by a scientist named as Aristotle. Alright, he classified plants into 3 categories, that is herbs, shrubs and trees. He classified all the plants into 3 categories, that is herbs, shrubs and trees. He also classified animals into 2 categories. One group which is having all the animals with no red blood cells, and another group of animals which is having all the animals with the red blood cells. So he classified animals on the basis of presence, or absence of red blood cells in them. Alright, but this system of classification is failed. This is failed. Why this is failed? Because he could not differentiate the all organisms, or he could not able to put all other organisms available or present at that time into different categories. So the system of classification given by Aristotle has been failed. Then a second scientist came into the system of classification. He introduced a 2-kingdom system of classification, and the scientist's name is Linnaeus. He proposed 2-kingdom system classification. Alright. So Linnaeus came and he proposed a 2-kingdom system of classification, and what he did, he actually classified all the animals and plants. He classified all the organisms, he made 2 groups, and he classified all the plants and animals into 2 groups, and he proposed 2 kingdoms, that is kingdom Plantae. He introduced kingdom Plantae, and he also introduced kingdom Animalia. This system of classification was also failed because this system was unable to differentiate between multicellular and unicellular organisms, and multicellular, unicellular organism and eukaryotes and prokaryote organisms. So this classification system was also failed. Then a scientist named as R.H. Whittaker has came and he introduced a 5-kingdom system of classification. He actually classified all the organisms into 5 different categories, 5 different kingdoms on the basis of some fundamental or characteristic features such as presence of cell wall, mode of nutrition, etc. So he chose some characteristics such as mode of nutrition, alright, then organization, cellular organization or body organization we can say. This was also a criteria to classify all the organisms into different categories, organization, then also presence of cell wall. So he classified all the organisms into different categories on the basis of these some characteristic features, and he gave 5 kingdoms, that is kingdom Monera, kingdom Protista, kingdom Fungi, kingdom Plantae, and kingdom Animalia. So this is a 5 kingdom system of classification which includes the 5 categories or 5 kingdoms, separate inside which or into which the different kind of animals or different kind of plants and different kind of organisms are placed into these categories. So we have finally 5 kingdoms, that is kingdom Monera, kingdom Protista, kingdom Fungi, kingdom Plantae and kingdom Animalia. This is known as 5 kingdom system of classification which was proposed or introduced by a scientist named as R.H. Whittaker. In the recent time right now we also follow this 5 kingdom system of classification. This is the most accepted system of classification given by R.H. Whittaker till now. So this is a complete solution of question number 1 here, and we can see where there are several changes which have been made into the system of classification. So this is a complete solution. Now we have question number 2 on the screen, that is state 2 economically important uses of A. Heterotrophic bacteria and B is Archaebacteria. So we have to state some economically important uses of these 2 types of bacteria. First we are going to talk about the heterotrophic bacteria. Heterotrophic means you all know that this is a type of mode of nutrition which is a type which includes all the organisms who feed upon different organisms, who obtain their nutrition from different organisms. They do not make their own food. All organisms except for plants, all the organisms present on earth are heterotrophic in nature. Only the plants available on earth are autotrophic in nature. That means they can synthesize their own food, but we cannot do so. So we are known as heterotrophic organism. If we are talking about bacteria, bacteria are also heterotrophic organisms because they are not capable of synthesizing their own food, hence they obtain their nutrition from other organisms. So we are talking about heterotrophic bacteria, and we have to state some economically importance of heterotrophic bacteria. So first economic importance of heterotrophic bacteria is they are actually helpful in decomposition. Bacteria are the decomposers and they help in the formation of humus. So first economic use of heterotrophic bacteria we are talking about. First of all we are talking about heterotrophic bacteria. Heterotrophic bacteria. Alright, and we are talking about the economic importance of these. So first economic importance of heterotrophic bacteria is they are decomposers. They are decomposers and they help in the formation of humus. Why so? Because they are decomposers. They feed upon these dead decaying matter also. So these kind of bacteria who feed upon dead or decaying matter are known as decomposers, and this is why they have economic importance, because they are helping in the formation of humus. If we talk about the another economic importance of heterotrophic bacteria, then we can say that some of the bacteria are useful in the formation of curd from milk. So this is one of the economic importance. Some bacteria such as Lactobacillus Bacteria, a special type of bacteria named as Lactobacillus Bacteria. This bacteria actually helps or converts the milk into curd. So these bacteria are also helpful in the formation of curd from milk, and hence they have economic importance of them. So second one is they help in the formation of curd from milk, and that is why they have economic importance. Alright, some of the species of bacteria are also helpful in the making of antibiotics, which are useful in the treatment of some diseases. Alright, so some of the antibiotics can also be obtained from some of the species of bacteria, hence they are useful, they are economically important to us. So third economic importance would be they are useful. They are useful in the formation of antibiotics, which can be obtained from some of the species of bacteria. Alright, and if we talk about some other kind of economic importance of bacteria, then these bacteria are also helpful in nitrogen fixing. So they can also fix nitrogen. Some of the bacteria such as cyanobacteria are very helpful in the fixation of nitrogen for large and advanced plants. So they are also helpful in nitrogen fixing. When they are in symbiotic relationship with advanced plants or large plants, they actually fixes nitrogen present in the atmosphere, and hence they have an economic importance in nitrogen fixing as well. So these are some economic importance of heterotrophic bacteria. Now we are going to discuss the economic importance of archaebacteria. Archaebacteria. Archaebacteria is a group of bacteria which includes all the bacteria who are resistant to extreme temperatures, who are adapted to survive in extreme of temperatures. Hence they are known as archaebacteria, and they are grouped under separate category of this archaebacteria. So archaebacteria have some economic importance. If we discuss the first economic importance, that they are actually helpful in production of methane gas. We know that methane gas is produced. First of all, methane gas is not produced from bacteria, it is produced from the dung. It is produced from the dung of ruminant animals such as cows, ruminant animals. Alright, so methane gas is produced from the dung of ruminant animals, and these archaebacteria such as methanogens, special kind of archaebacteria known as methanogens, are found in the gut of ruminant animals. They are also found in, hence they are found in dung of the ruminant animals. So biogas is produced with the help of methanogen bacteria, and these methanogen bacteria are actually they fall under the category of archaebacteria, methanogen bacteria. So they produce biogas, they produce methane gas, and also these archaebacteria help in sewage treatment. They are used in sewage treatment. Alright, so these are the economic importance of archaebacteria here. We have already discussed the economic importance of both of bacteria such as heterotrophic bacteria and archaebacteria. So this is the complete solution of question number 2 on the screen. Now on the screen we have question number 3, that is, what is the nature of cell walls in diatoms? So first of all, what is diatoms? What is diatoms? Diatoms are algae. Diatoms are algae. So diatoms are a kind of algae that dwells in glass houses. They are the only organisms with having cell walls made up of transparent opaline silica. If we talk about the nature of cell walls of diatoms, then the cell walls of this diatom algae actually arranged like this and forming a soap-like box. This is the cell wall arranged like this, cell walls of diatoms arranged like this. They are forming a soap box and two overlapping cells. The cell wall of diatoms are actually overlapping on each other and forming two cells like this. These are the two thin cell walls of diatom algae. And this cell wall of diatom is actually made up of silica, as we already have mentioned that these are the only organisms, diatoms are the only organisms which have cell wall made up of the transparent and opaline silica. So this is the nature of cell wall of diatoms, that they are arranged like this, thus they are forming two overlapping cells and they are made up of silica. So if you want to write, then first of all, diatoms are algae. Diatoms are algae that dwells in glass houses. And they are the only organisms. They are the only organisms which have cell walls made up of transparent opaline and opaline silica. Hence they are not destructible also, because they are having the cell wall made up of silica, so they are not destructible. They are non-destructible. Alright. If we talk about the nature of cell wall of diatoms, then the cell walls of diatoms forms two overlapping cells, like this, and the cell wall of diatoms is actually made up of silica. It is made up of silica, and hence they are non-destructible because of the presence of silica in their cell wall. They are non-destructible. So this is the nature of cell wall of diatoms, and diatoms are actually algae. And this is the complete solution of question number 3. Now we are going to discuss question number 4. Find out what do the terms algal bloom and red tides signify. So first we are going to talk about the algal bloom. Algal bloom term refers to the increase in number of algae or blue-green algae, that is cyanobacteria in water. So algal bloom refers to the increase in number of algae or cyanobacteria in water. This increase in number of cyanobacteria or blue-green algae in water actually discoloration, this provides or causes discoloration of the water body. This also increases the biological oxygen demand. When the number of cyanobacteria or blue-green algae increases in water body, this increases the biological oxygen demand, which can lead to the death of large number of fishes present in the water body as well as some other aquatic animals. Why this is so? Because if we are talking about the algal bloom is just a rapid increase in the number of blue-green algae or cyanobacteria. If the number of cyanobacteria or blue-green algae is being enlarged or being increased in the water body, they will feed on the organic matter present in water body. And if they are feeding in the water body on the organic matter, they will require more oxygen. This increases the demand of biological oxygen demand. This is known as biological oxygen demand, BOD. And this causes the death of fishes and other aquatic animals present in that particular water body. So this is known as algal bloom. If we talk about the second term given in the question, that is red tides, then this red tide caused by dinoflagellates, such as goniolax, and this, because of the large number of goniolax or dinoflagellates, the sea appears red in color. The sea appears red in color in case of red tides, and the water or any water body inside which the blue-green algae multiplies and increase their number, that water body becomes green in color. And in case of red tides, the sea becomes red in color because of the increase in number of dinoflagellates such as goniolax. These red dinoflagellates or goniolax actually releases some toxic substances or toxin chemicals in the water body or sea, due to which the large number of fishes and other aquatic animal actually dies. So this is red tide and this is your algal bloom. If you want to write the answer, then first we are going to talk about the algal bloom. Algal bloom. So algal bloom is, the term refers The term refers to the increase in number of blue-green algae, in water body. Basically, they reside on the surface of any water body. That is why the water body looks green in color. So the term algal bloom refers to the increase in number of blue-green algae. The water body hence looks green in color, because of large number of algae present here, and this also increases BOD. This also increases BOD. Now what is BOD? Biological oxygen demand. This also increases biological oxygen demand. And because of this increased biological oxygen demand, most of the fishes present in the water body, because of this increased biological demand, most of the fishes and other aquatic animals dies. Alright, so this is algal bloom. If we talk about the red tide, so this causes or caused by red dinoflagellates, such as goniolax, So this caused by red dinoflagellates such as goniolax, and because of increased number of these dinoflagellates, the sea appears red, because the dinoflagellates are red, appears red, and they release toxic substances into the sea, which leads or results into the death of large number of fishes, and also some other aquatic animals present in that sea. So this is the basic difference between algal bloom and red tide, and we have already discussed what is algal bloom, what is red tide. Algal bloom is caused by cyanobacteria, or we can say the blue-green algae, which increases biological oxygen demand, and this increased biological oxygen demand actually leads to the death of fishes present in that water body. And the red tide is caused by dinoflagellates, red dinoflagellates specifically, and this goniolax, This goniolax is actually a member of red dinoflagellates, which is responsible for causing red tides in the sea. the sea actually appears red in color because of the large number of red dinoflagellates in sea. These red dinoflagellates releases toxic substances which lead or result into the death of a large number of fishes or aquatic organisms present in that sea. So this is a complete solution of question number 4. Now we are going to discuss question number 5. How are viroids different from viruses? So we have to basically differentiate between viruses and viroids here. So first of all, viroids. What are viroids? Viroids are infectious entities which can affect plants. Viroids are actually infectious entities which can affect plants, and they are smaller in size as compared to the viruses. If we talk about viruses, what are viruses? Viruses also are infectious agent with only nucleic acid molecules surrounded by a coat of protein. They are not cells and they are not considered as living as well, because they actually have a dual life. When they are in the environment, they act as they are dead organisms, and when they enter into the host organism, when they enter into another organism such as in plants, in case of animals, they become active. They act as a living molecule. So viruses are not considered as living. And what are viruses? Viruses are just nucleic acid molecules surrounded by a coat of protein, and they are infectious. If we talk about the sizes of viroids and viruses, viroids are smaller as compared to the viruses. Alright, now if we want to discuss the differences between viroids and viruses, then viroids have only single circular DNA, whereas viruses having numerous DNA. They are having numerous DNA. In case of viroids, RNA doesn't code for proteins. In a normal case of any living organism, we know that RNA codes for protein. Alright, but in case of viroids, RNA doesn't code for protein, but in case of viruses, RNA codes for protein. RNA codes for protein in case of viruses. Viruses also have numerous. They are having numerous DNA, whereas viroids have only a single circular DNA. If we talk about the RNA copying in case of viroids, RNA copied in viroids in only nuclear regions. And in case of viruses, the RNA copied into cytoplasm as well as in nuclear regions. So these are the basic differences between the viroids and viruses. So we are going to write the answer on screen here. First of all, we have to give definition what is viroid and viruses. So first we are talking about viroids. Viroids. These are infectious entities. That means agent, we can also call them agent. These are infectious entities which can affect plants, and they are smaller in size as compared to the viruses. If we talk about the viruses, they are also infectious agent. They are also infectious agents. They cause infection to both plants and animals. So they are also infectious agents with only nucleic acid molecule surrounded by a coat of protein, surrounded by a coat of protein. Alright, so these are the definitions of viroids and viruses. They are not having protein coats. Viroids lack protein coats and they have protein coats, viruses have protein coats. If we want to differentiate between viruses and viroids, then here we are going to discuss viruses and here we are going to write about viroids. Alright, so first of all, RNA in case of viroids doesn't code for protein, whereas in case of viruses RNA codes for protein. RNA codes for protein in case of viruses. Then viroids have only single circular DNA. They have only single DNA, whereas viruses have numerous DNA are present in viruses. Numerous DNA they are having. Then RNA copying occurs only in nucleus, and in case of viruses it can happen in cytoplasm as well as nucleus. Alright, if we talk about the size, then they are comparatively smaller. As compared to the virus's size. So this is a complete solution of question number 5 on the screen. So now we are moving on to question number 6, that is describe briefly the 4 major groups of protozoa. So first of all, what is protozoa? Protozoa is a group of members which are unicellular in nature and are called protists. So these protozoas are unicellular protists. They exhibit heterotrophic mode of nutrition. They can be holozoic, they can be parasitic, as well as they can be saprophytic in nature. These all kind of nutritions, saprophytic, holozoic, parasitic nutrition, all these type of nutritions are included in heterotrophic nutrition. They are not autotrophic at all. So these protists, all the protists are not autotrophic, all the protists are heterotrophic in nature, and the protozoans are basically divided into 4 major groups such as amoeboids or known as sarcodines. Then we have flagellated protozoans or known as flagellates. Then third we have is ciliated protozoans or known as ciliates, and the last 4th category or group of protozoa we have is sporozoans. So if we talk about the very first category of protozoans, that is amoeboids or sarcodines, this group of protozoa includes amoeba and ant-amoeba. So basically the group of amoeboids or sarcodines are unicellular in nature. They do not have flagella or cilia present in their body. They lack flagella and cilia. Instead of that, they have a characteristic feature known as pseudopodia. They have protoplasmic outgrowths which are known as false feet and are known as pseudopodias as well. So these pseudopodias are the characteristic features of the members of amoeboids or sarcodines, and these pseudopodias help these organisms to move and capture their prey. They may be free living and they may be parasitic in nature. If we talk about the free living member of amoeboids or sarcodines, that is amoeba, and the parasitic form of sarcodines is ant amoeba. So this is all about the amoeboids or sarcodines. Moving on to the next category or group of protozoa, that is flagellated protozoa. We have a group of some organisms included in the flagellated protozoa because their body consists of flagella. Their body consists of flagella. They lack cell wall. They do not have cell wall. Their body consists of flagella, and the presence of flagella is actually the characteristic feature of the members of flagellated protozoans. That is why the group has been named as flagellates or flagellated protozoans. These flagellas are present in them, helping them to move as well as capturing in the prey. A common organism, parasitic organism, which is parasite in nature, known as Trypanosoma. Trypanosoma is included in the category of flagellated protozoan because it is having flagella, and this Trypanosoma is actually a responsible agent to cause sleeping sickness in humans. Sleeping sickness is a disease which is caused by this Trypanosoma included in the flagellated protozoans in the humans. Then third category of protozoan we have is ciliated protozoans, and this ciliated protozoans are the organisms who bear numerous cilia on their body surface. Numerous cilia are present on their body surface. These all cilia beat in one direction to move water laden food into their cavity known as gullet. Alright, so next we have is sporozoans. Sporozoan is the last category or group of protozoa into which all the endoparasites, all the disease causing agents are included in this category, sporozoans. This also includes malarial parasite, malarial causing agent known as plasmodium. So plasmodium is spore, it is a sporon, it is sporozoan, and it is a protozoan which is included in the category of a protozoan known as sporozoan. Now if we are going to write the answer, then first of all, protozoa are unicellular protist, unicellular protists exhibiting heterotrophic mode of nutrition. This can be holozoic, this can be parasitic, as well as this can be saprophytic. These protozoans are divided. These protozoans are divided into four major groups, What are these? These are amoeboids. Second one is flagellated protozoans. Third is ciliated protozoans. And the last one we have is sporozoans. Amoeboids or sarcodines include the member which are unicellular in nature. So these are unicellular organisms. They do not have flagella as well as cilia in them. Instead of that, they have pseudopodia. Which is nothing but outgrowth of protoplasm of them. Pseudopodias are the outgrowth of protoplasm of amoebas or the organism included in the amoeboids or sarcodines. These pseudopodias help these organisms to move and in capturing of prey. So these pseudopodias help them to move and capturing of prey. They can be free living or they can be parasitic in nature. If we are talking about the free living one, free living example is amoeba, and the parasite. Parasitic form we have is ant amoeba. Moving on to the next category, that is flagellated protozoans. They are having flagella. They have flagella. That is why the group has been named as flagellated. So they have flagella which helps them to move and capturing in prey. Then these leg cell wall also, they can be free living, they can be parasitic in nature, and a very famous example, that is Trypanosoma. Trypanosoma is the member of flagellated protozoan which is responsible for causing sleeping sickness in humans. The next category we have a ciliated protozoan, and in this all the organisms with having numerous cilia are included in the category of ciliated protozoans. So their body is covered. With numerous cilia, all these cilia beat in one direction to move water laden food into the cavity called gullet, And the last category of protozoans, that is sporozoans, this include all the pathogens and endoparasitic forms. So this include all the endoparasites, plus pathogens. Pathogens are those agents which is capable of causing disease into other organisms. All these are known as pathogens. So this category, this last category of protozoan, that is sporozoans, including all the endoparasites and pathogens. This also includes malarial pathogen known as Plasmodium. So I hope it is clear now, and this is all about question number 6. This is a complete solution and answer of question number 6. Now moving on to the question number 7. Plants are autotrophic. Can you think of some plants that are partially heterotrophic? Yes, obviously plants are autotrophic. Most of the plants, almost all, not all, almost all plants are autotrophic in nature. That means they are having chlorophyll pigment in their chloroplast, which is responsible for the process of photosynthesis. Now what is photosynthesis? We all know this is a process of synthesizing food, and this property of synthesizing food is known as autotropism, which is the characteristic feature of only plants. Because of the presence of photosynthesis and because of the presence of chlorophyll pigment in their chloroplast, plants are known as autotrophic organisms. No other organism on earth, no other organism is capable of synthesizing their own food. Only the plants are the organisms which are capable of synthesizing their own food, hence they are known as autotrophic organism. But some of the plants or some of the species of plants are heterotrophic. They are partially heterotrophic. Which one of them? Some insectivorous plants are partially heterotrophic. That doesn't mean that they are not autotrophic. They are autotrophic. All the plants are autotrophic, but some insectivorous plants who eat upon insects to get their nutrition are partially heterotrophic. That means some plants or insectivorous plants play dual mode of nutrition. They are autotrophic and they are heterotrophic as well. They can synthesize their own food when they do not obtain proper or appropriate amount of nutrients. They can switch on to another type of mode of nutrition, that is heterotrophic nutrition. Insectivorous plants eat insects. They have various means of capturing insects to obtain, so as to supplement their diet. They actually eat insects, or they eat upon insects to supplement their diet with the required amount of nutrients obtained from insects, and by they are causing the proliferation in their growth of insects. So some insectivorous plants are autotrophic in nature, but they can switch on to heterotrophic mode of nutrition. They eat insects. They have various means of eating or capturing insects. Various kind of plants, various kind of insectivorous plants have various means to capture insect so as to supplement their diet with the required amount of nutrients obtained from insects by causing proliferation in the growth of insects. We have some examples of insectivorous plants such as pitcher plant, Venus flytrap, bladderwort and sundew plants. So this is answer of question number 7, and if you want to write, then it will goes like this. Yes, plants are autotrophic in nature. They are autotrophic. We all know that plants are autotrophic in nature because they are capable of performing photosynthesis. They are capable of performing photosynthesis. And why they are capable of performing photosynthesis? Because they have chlorophyll pigment, a special type of pigment present in them. They have chlorophyll pigment which is not found in other members except for algae or blue-green algae. So blue-green algae, cyanobacteria, algae are capable of synthesizing food, but except for plants and algae, any member on the earth do not have chlorophyll pigment, hence they are not capable of producing or synthesizing food by own. So they have chlorophyll pigment, in their chloroplast, So they are known as autotrophic organism, but some heterotrophic plants we have, that is insectivorous plants. Alright, they have various means of capturing insects. They have various means of capturing insects they have. Why they do so? So they can supplement their diet, with required nutrients obtained from insects, alright. They capture insect, they eat upon insect to supplement their diet with required nutrients obtained from insects, causing proliferation in their growth, So they are partially heterotrophic. Some insectivorous plants we have, they are autotrophic. Yes, they are autotrophic, but they are heterotrophic as well. They can switch on to another type of mode of nutrition, that is heterotrophic nutrition, when they are not having appropriate amount of nutrients, and they can eat insects, they can eat upon insects to supplement their diet with required amount of nutrients, obtaining from insects. We have some examples, common examples of insectivorous plants which are autotrophic in nature as well as they are heterotrophic in nature. That means they eat upon insects. So first, if we talk about, they have pitcher plant. We have common example of insectivorous plants. Then we have Venus fly trap. We have bladderwort, and the last one we have is sundew plant. So all these are the examples of insectivorous plants. These insectivorous plants are not at all heterotrophic, they are partially heterotrophic. They are autotrophic as well. They can switch on to autotrophic and heterotrophic as well, depending upon their requirement of nutrients. So this is a complete solution of question number 7. Moving on to the question number 8. What do the terms phycobiont and mycobiont signify? So we have two terms in this question and we have to identify what these terms actually means. So phycobiont term refers to the algal component in lichen, and mycobiont refers to the fungal component of lichen. Now what is lichen? Lichen is actually a symbiotic relationship between fungus and alga. These are living in the association with each other and making a symbiotic relationship. Now, what are the functions of this symbiotic relationship? As we know that this symbiotic relationship is between an alga and fungus. So, both are doing their specific functions. Algae produces or synthesizes food because of the presence of chlorophyll in them. They prepare food for fungus, and fungus absorbs nutrient and water from soil and provides it to the alga. Also, it provides the shelter for algae. So this is symbiotic relationship of algae and fungus, in which we have two components, algae and fungus. Algae is known as phycobiont and fungus is known as mycobiont. If we write answer, it would be like this, that phycobiont refers to the algal component of lichen. Lichen is the word for the symbiotic relationship of algae and fungus. And mycobiont refers to the fungal component of lichen. As I have already mentioned what is lichen. Now here we are going to write lichen is the symbiotic relationship or association, or we can call it a relationship. This kind of relationship in which the algae and fungus are included, both are benefited by each other. That is why the relationship between these two are known as symbiotic association or relationship. Lichen is a symbiotic association or relationship which includes phycobiont as algae and mycobiont as fungus. In this relationship or symbiotic association, the algae produces or synthesizes food for algae, food for itself, and algae also, fungus also food for itself and fungus as well, and on the other hand, what does fungus do? Fungus provides shelter to algae, also it absorbs water plus nutrient from soil for the growth of algae. So this is the complete solution of question 8. That I hope you are clear with the answer of question number 8. What is phycobiont and what is mycobiont? Phycobiont is actually the algal component of lichen, and mycobiont is the fungal component of lichen. Lichen is a symbiotic association or relationship between fungus and an algae. They too have different specific functions for each other. This kind of relationship is a benefited relationship in which both the components, both the organisms, fungus as well as algae, are benefited by each other. Algae is providing food to the fungus and fungus is providing shelter and nutrients and water from soil to the algae. So this is a complete answer of question number 8. Now on the screen we have question number 9, and we have to give a comparative account of the classes of kingdom fungi under the following, that is mode of nutrition and mode of reproduction. So in this question basically we have to give a comparative account of different classes of kingdom fungi with respect to the mode of nutrition in them as well as mode of reproduction in them. Basically we have 4 classes, 4 different groups of kingdom fungi, and this includes Phycomycetes, Ascomycetes, Basidiomycetes and Deuteromycetes. So basically we have 4 classes of kingdom fungi. First one is Phycomycetes, then we have Ascomycetes, Deuteromycetes and Basidiomycetes. First, if we talk about the first class of kingdom fungi, that is Phycomycetes. This group of fungi, Phycomycetes, is a group of fungi which includes organisms like Rhizopus, Albugo, etc. These Rhizopus and Albugo are example of Phycomycetes. If we talk about the mode of nutrition in them, they are obligate parasites. They are obligate parasites on plants. And if we talk about the reproduction, mode of reproduction in Phycomycetes, they can reproduce asexually and they can reproduce sexually as well. If we talk about asexual reproduction in Phycomycetes, they produces motile spore. They can reproduce asexually with the help of motile spores known as zoospores. They also produces non-motile spores. They also reproduces by asexual means with the help of non-motile spores known as aplanospores. If we talk about sexual reproduction in Phycomycetes, yes, they can reproduce sexually, and the sexual reproduction in Phycomycetes can be of isogamous, oogamous and anisogamous as well, which result into the formation of thick-walled zygospores. So this is all about Phycomycetes. Now we will write first class of kingdom fungi, that is Phycomycetes here. As we have discussed the very first class of kingdom fungi, now we are going to write about the features as mode of nutrition and mode of reproduction of Phycomycetes. So Phycomycetes, first of all, is a group of fungi which includes Rhizopus, albugo, etc. These are the examples of Phycomycetes. If we talk about the mode of nutrition, of Phycomycetes members, they are obligate parasites. On plant. That means they are heterotrophic, and in heterotrophic they are parasitic in nature. They exhibit heterotrophic mode of nutrition. They are not capable of synthesizing their own food because they are not having chlorophyll pigment in them. Hence they are heterotrophic in nature. And what kind of heterotrophic they are? They are obligate parasites on plants. If we talk about the mode of reproduction, we know that the members of Phycomycetes can reproduce asexually as well as sexually. If they are reproducing asexually, they produces or reproduces with the help of motile spores, known as zoospores, and sometimes they reproduces through non-motile spores known as aplanospores. The important thing to remember is these both type of spores, zoospores which are motile in nature and aplanospores which are non-motile in nature. These both type of spores are produced in the special structure in them which is known as sporangia, and they are produced inside the sporangium. That is why we can call them that these both spores, zoospores and aplanospores, are produced endogenously in a specialized structure known as sporangia. If we talk about the sexual means of reproduction in them, it can be isogamous, it can be anisogamous, or it can be oogamous, which result into the formation of thick-walled zygospore, which result into the thick-walled zygospore. So this is about Phycomycetes. Now we already know that the fungi has been classified into 4 different categories such as Phycomycetes, Ascomycetes, Basidiomycetes and Deuteromycetes. We have discussed Phycomycetes, which is the first class of fungi. Then we are going to discuss now the second class of fungi, that is Ascomycetes. Now this class of fungi such as Ascomycetes includes members like Penicillium, Aspergillus, Claviceps and Neurospora. These are the members which are included in the second class of fungi, that is Ascomycetes. If we talk about the mode of reproduction in Ascomycetes, they can be saprophytic in nature, they can be decomposer, they can be parasitic as well, and also they can be corpophilous. Now what does that mean? What does corpophilous means? The organisms which can grow or feed upon dung, those organisms are known as corpophilous. So this is all about the nutrition type of Ascomycetes. If we talk about the reproduction, mode of reproduction in Ascomycetes, then the members such as Neurospora, Claviceps or all the members which are included in Ascomycetes can reproduce through both the means of reproduction, that is asexual reproduction and sexual reproduction. They can reproduce asexually with the help of some spores such as conidia in conidiophores, which are produced exogenously on them. If we talk about the sexual means of reproduction in them, they can reproduce through Esco Spores. They can reproduce through Esco Spores, which is a type of sexual reproduction in them. These Esco Spores produced endogenously in them. That means the spores, Esco Spores, which are sexual spores, are produced inside their body, and these are produced in a sac-like structure called as asci and are arranged in ascocarps. So this is all about the mode of nutrition and reproduction mode in Ascomycetes, which is the second class of fungi. If we want to write the second class of fungi, that is Ascomycetes. Alright, in Ascomycetes, first of all we will write some examples. We will write examples. What are them? They are Penicillium. The Penicillium which is used to make antibiotics. We know that the first antibiotic was prepared with the help of penicillium. This penicillium, that penicillium is actually included in the class of Ascomycetes. So under this Ascomycetes, which is the second class of fungi, we have Penicillium. Then we have Aspergillus. Aspergillus. Also we have Claviceps and Neurospora. So all these are the examples of Ascomycetes class of a fungi. Then if we talk about the mode of nutrition in them, the mode of nutrition in them can be saprophytic in nature. They can be saprophytic. They can be saprophytic in nature. They can be decomposer. They can be decomposer. They also can be parasitic. They also can be parasitic. That means they can stay and they can obtain their nutrition from the host organism as well. And also they can be corpophilous. Corpophilous, which means they can grow and feed upon dung. Alright, so they can feed upon dung also. So these are the mode of nutrition in class of Ascomycetes. If we talk about the mode of reproduction in them can be asexual, and sexual as well. If we talk about the asexual means of reproduction in Ascomycetes, they can reproduce through spores. known as conidia, which is very common in case of fungi. So conidia are asexual spores produced in Ascomycetes members of fungi. These conidia, which are asexual spores, are produced exogenously. That means not inside the body of them. The spores known as conidia, which are asexual spores, are actually produced outside the body of organism or fungi. That means exogenously. Alright, if we talk about the sexual means of reproduction. Sexual means of reproduction, they also reproduce by sexual means by producing sexual spores, that is Esco spores. Esco spores are the sexual spores produced inside the body of Ascomycetes members. That means Esco spores are going to produce endogenously because they are producing inside the body. So these spores are produced endogenously, alright, but inside the special structure, which is a sac-like structure known as asci. So these Esco spores are going to produce inside asci and are arranged in ascocarps. So this is all about the second class of fungi, that is Ascomycetes. Now we already have discussed the examples or the members which are included in the class of fungi, that is Ascomycetes. We already discussed the mode of nutrition in the members of Ascomycetes, and also we have discussed the reproduction, which is asexual and sexual. They can reproduce by both means of reproduction, that is asexual and sexual, and we have discussed the different types of reproduction in them. That is, if we talk about asexual, they can produce or they can reproduce with the help of asexual spores known as conidia, which are very common in them, and these conidias, which are asexual spores, are produced exogenously, that means outside their body. And if we talk about the sexual means of reproduction, they also can reproduce sexually by producing sexual spores known as Esco spores, which are produced inside their body in a sac-like structure known as asci and are arranged in ascocarps. So this is all about the second class of fungi, Ascomycetes. Now here we are going to talk about the third class of fungi, that is Basidiomycetes. Now this Basidiomycetes, which is a third class of fungi, includes some members such as Eustylago, Agaricus and Puccinia. All these are the examples or the members which are included in the class of fungi, that is Basidiomycetes. If we talk about the mode of nutrition in Basidiomycetes, then they can be decomposers in soil, they can be decomposers on tree logs and tree stumps. So they can be decomposers, alright, they can be decomposer in soil, they can be decomposer on tree logs and tree stumps. They also can live as parasite in some plants. They can also live as parasite in some plants. That means they can obtain their nutrition from that plants inside which they are living. And they also can cause some diseases such as rust and smut in those plants. So this is all about the mode of nutrition in the members of Basidiomycetes. That means they can be decomposers in soil, tree logs and tree stumps, and they also can live as parasite in some plants and also can cause some diseases such as rust and smuts. Now if we talk about the reproduction, mode of reproduction in the members of Basidiomycetes, then if we talk about the asexual reproduction, we have basically two types of reproduction, that is asexual reproduction and sexual reproduction. Then if we talk about the asexual means of reproduction in the members of Basidiomycetes, there can not be produced asexual spores. They cannot produce asexual spores. Hence, they are not capable of reproducing through asexual means because there are no spores are going to produced in them which are asexual in nature. So they cannot reproduce asexually, but they can reproduce by the vegetative means of reproduction, which is also a type of asexual reproduction, and they can reproduce by the fragmentation method of vegetative reproduction. We know that the difference between sexual reproduction and asexual reproduction, that asexual reproduction does not involve the formation and fusion of gametes, whereas sexual reproduction is characterized by the fusion of two different types of gametes. Similarly, vegetative reproduction does not involve any fusion of gametes. That is why this is considered as a type of asexual reproduction. But the thing to remember in the members of Basidiomycetes is, we know that in the previous classes of fungi, we know that the asexual spores are produced, such as conidia, through which the Ascomycetes were going to reproduce by asexual means. But in the members of Basidiomycetes, they are not going to produce asexual spores. There is no production of asexual spores, hence they cannot reproduce by the asexual means of reproduction, but they can reproduce through vegetative means of reproduction with the help of fragmentation. That means their body can break down into different fragments, and each fragment can give rise to a new member of Basidiomycetes. This is all about the asexual reproduction. If we talk about the sexual means of reproduction in Basidiomycetes, then the sex organs are also absent in the members of Basidiomycetes. They are not having any kind of sexual organs in them. So then how could they reproduce? How the reproduction takes place in the members of Basidiomycetes? If they are not reproducing through asexual means, they are undergoing fragmentation for their reproduction. And when it comes to sexual reproduction, the sex organs are also absent in them. Alright, they cannot reproduce asexually, they cannot reproduce sexually because they are not having any sex organs, but a similar kind of sexual reproduction can happen in them, that is known as plasmogamy. They can reproduce through plasmogamy, which is considered as sexual reproduction in the members of Basidiomycetes. Now what is plasmogamy? Plasmogamy involves the fusion of two different type of strains of hyphae. This results into the formation of a dikaryon stage, and this dikaryon stage will produce basidium, inside which the 4 basidiospores are going to produced. So this is how the members of Basidiomycetes class of fungi actually reproduces. They do not reproduce through production of asexual spores or asexual means of reproduction. They can reproduce through fragmentation, which is a means or a method of vegetative reproduction. If we talk about the sexual reproduction, they can reproduce through plasmogamy, which involves the fusion of two different types of strains, which result into the formation of a dikaryon stage, and which also result into the formation of a basidium, and finally going to produce basidiospores inside the basidium. So we are going to discuss about Basidiomycetes. As we know that, first of all, we are going to write examples or the name of those members which are included in the class of Basidiomycetes of fungi. Then we know that Eustylago, Agaricus and Puccinia, which is responsible for causing diseases in plants because they are living as parasites in them. Now, if we talk about the members of Basidiomycetes, we know that Eustylago, Agaricus and Puccinia are the members which are involved in the class of fungi that is known as Basidiomycetes, which is the third class of fungi. If we talk about the mode of nutrition in them, if we talk about the mode of nutrition, they can be decomposers. They can be decomposers in soil. They can be decomposer in soil on tree logs and tree stumps So this is the nutrition, one type of nutrition which can be exhibited by the members of Basidiomycetes. They can be decomposers in soil on tree logs and tree stumps. Also they can live as parasite on plants, parasite on some plants, and can cause some diseases such as rust and smut. Some diseases such as rust and smut. So this is all about the mode of nutrition exhibited by the members of Basidiomycetes. Last, we are going to talk about the mode of nutrition in them, mode of reproduction in them. Then there is no production of asexual spores can be found in the members of Basidiomycetes. So no asexual spores are produced in Basidiomycetes, hence they can reproduce through vegetative reproduction and by the method of fragmentation, which is a type of method included in the vegetative reproduction only. So they can reproduce fragmentation, that means the body can be divided into different fragments and each fragment can give rise to a new fungi. If we talk about the sexual means of reproduction, they can reproduce through plasmogamy because they are not having sexual organs on them. So they can reproduce through plasmogamy, which is a type of sexual reproduction inside which this is a process basically of reproduction. What happens in the plasmogamy? The two different types of strains of hyphae actually fuses with each other. Hyphaes are haploid in nature. All the hyphaes as the body are haploid. So hyphaes are haploid in nature. The two haploid hyphaes, the two different strains of hyphae which are haploid in nature are going to fuse with each other to form a dikaryotic stage, and this dikaryotic stage is going to produce basidium, which is going to produce 4 basidiospores inside the basidium. So this is all about the Basidiomycetes or the members of Basidiomycetes. We already have discussed the nutrition, how they actually take or obtain their nutrition by different ways, and also we have discussed how they actually reproduces. That means they can reproduce through fragmentation and plasmogamy. So this is all about the answer of this. Now we are going to move the last class. Now we are going to discuss the last class of fungi, that is Deuteromycetes. So now let's discuss the last class of fungi, that is Deuteromycetes. And if we talk about the members of Deuteromycetes, then Alternaria, Trichoderma and Colatotrichum, all these are the examples of Deuteromycetes or are the members which are included in the class of Deuteromycetes. Alternaria, Trichoderma and Colatotrichum. If we talk about the mode of nutrition in the members of Deuteromycetes, some of the members of Deuteromycetes can be saprophytic, some members can be parasitic in nature, and a large number of Deuteromycetes are actually decomposers on leaf litters. Now this is the reproduction, this is the type of nutrition they can be exhibited by the members of Deuteromycetes. If we talk about the mode of reproduction in them, so simply they can reproduce by asexual means of reproduction only, such as with the help of conidia, which are asexual spores. And if we talk about the sexual means of reproduction, that is absent in them. That means the members of Deuteromycetes cannot reproduce sexually, they only can reproduce through asexual means with the help of production of asexual spores such as conidia. The thing to remember is asexual spores. Asexual spores such as conidia are very common, and most of the fungi reproduces through conidia, which are the asexual spores. So last class of fungi we have is Deuteromycetes. First we are going to talk about the examples or the members which are included in the class of fungi, that is Deuteromycetes. So first we have is Alternaria. Second is Trichoderma. And the last is Colatotrichum. These all three are the members or examples which are included in the class of Deuteromycetes of fungi. Now if we talk about the mode of nutrition in the members of Deuteromycetes, then some members of Deuteromycetes are saprophytic in nature. Some members can be saprophytic in nature, and some members of Deuteromycetes can be parasitic in nature. That means they can obtain their nutrition by living inside the host organism. And a large number of organisms included in the class of Deuteromycetes are actually decomposers. Large number of organisms included in the class of Deuteromycetes are actually decomposers on leaf litters. If we talk about the reproduction type in them, they can only reproduce through asexual means of reproduction with the help of very common spores, that is conidia. Only asexual reproduction can be found in them, and they reproduce with the help of very common spores which are asexual in nature, that is conidia, and sexual reproduction is actually absent in them. That means they cannot reproduce through sexual reproduction. Only they can reproduce through asexual means of reproduction with the help of very common spores such as conidia. So now we have completed with this fourth and last class of fungi, we have completed the answer of question number 9, which includes 4 classes of fungi, that is Phycomycetes, Ascomycetes, Basidiomycetes and Deuteromycetes. We have discussed the mode of nutrition and reproduction type, or the mode of reproduction in all the members or all the classes of fungi. With this, we have completed the question 9, and this is the complete solution of this question. Now let's come towards the next question, that is question number 10. What are the characteristic features of euglenoids? Now here we are going to talk about the characteristic features of euglenoids. First of all, if we are talking about the characteristic features of euglenoids, we have to know that what are basically euglenoids and where they are classified into. So euglenoids basically are known as euglena, which are unicellular in nature. That means they are composed of only one cell. They are unicellular bodied organisms. Euglena is a unicellular organism which is included in the Protista kingdom. Euglenoids such as euglena are unicellular in nature and included in the kingdom Protista. They can be found commonly in fresh water. Also, if we talk about some other characteristic features of euglenoids such as euglena, then they do not have cell wall. The cell wall is actually absent in the members of euglena. or in the euglena, instead of which they have a protein rich layer, protein rich cell membrane, which is known as pellicle. So they do not have cell wall, instead of which they have a protein rich cell membrane. Also they have a light sensitive eye spot on their body. This is one of the characteristic feature of euglena. Also they have two flagellas present on the anterior part of their body. Alright, so all these are the characteristics, and the last but not least, they have chlorophyll pigment in their body. They have a pigment which is known as chlorophyll, which is similar to the plants, and hence because of the presence of chlorophyll pigment in their body, they are capable of synthesizing food of their own, by their own. But in the absence of light, they can behave similar to heterotrophs. That means in the absence of light, they can obtain their nutrition by feeding on some other aquatic organisms present in that fresh water. They have a dual mode of nutrition. This simply means that they have dual mode of nutrition, because they are having chlorophyll pigment in their body, by which they can synthesize their own food in the presence of light. But if the light is not present in them, for them, they can switch on to another mode of nutrition, that is heterotrophic mode of nutrition. They can feed upon other aquatic small organisms present nearby in the fresh water. This property, because of the presence of some properties which are similar to the plants and some properties which are similar to the animals, this actually makes them difficult to classify into different categories, or actually it is very difficult to classify whether the euglenoid or euglena is a plant or a animal. So that is why they are known as connecting link between plants and animals. So if we want to write the answer of question number 10 or the characteristics, so the characteristics are as follows, and we are talking about the characteristics of euglenoids. So the first characteristic of euglenoids is the euglenoids such as euglena is unicellular. That means the body is actually composed of only one cell. They are unicellular in nature and they can be found in fresh water. Next thing, they having two flagella. They're having two flagella on the anterior portion or anterior part of their body. So they are having two flagella on the anterior portion of their body. Also, they do not have cell wall. They do not possess cell wall. So cell wall is absent in them. Instead of which they have a protein layer, a protein rich layer. They have a protein rich layer which is known as cell membrane. That means their cell membrane is actually protein rich. Instead of which they have a protein rich cell membrane. Alright, so they are not having cell wall, which is a feature of plants. Instead of which, they are having protein-rich cell membranes. So we can conclude that they are animals because of the presence of cell membrane only and the absence of cell wall. We can consider them as animal. But they are also having some features similar to the plants, that is the presence of chlorophyll. So if we talk about, first of all, if we talk about the fourth feature, they have a light sensitive eye spot, and as I already mentioned that they have a feature similar to the plants, that is they have a pigment which is known as chlorophyll. So they have chlorophyll pigment which is similar to the plants, and because of the presence of chlorophyll pigment, they can synthesize their own food, and if the light is not present for them, if in the absence of light, they can switch on to another mode of nutrition, that is heterotrophic nutrition. And from here we can conclude, because of the presence of chlorophyll pigment in them, we can consider them as plant. But on the other hand, we know that they do not have cell wall, and the main characteristic features of a cell of plant is they have a cell wall. So we cannot consider, or we cannot clearly classify the euglenoids into plants, whether they are belonging to the plants or whether they are belonging to the animals, because they are having the features which are similar to animals, they are having also the features which are similar to plants. So they are having chlorophyll, and with the help of chlorophyll they can synthesize their own food. In the absence of light, they can switch on to another mode of nutrition, that is heterotrophic nutrition. That means they can obtain their nutrition by feeding upon some other aquatic organisms present in the fresh water. And because of the presence of both the features similar to animals and plants, this is difficult for us or scientists to classify them into a particular category. So that is why they are put into the kingdom Protista, and they are considered as the connecting link between plants and animals, because they possess the features similar to both of the plants and animals. So these are the characteristic features of euglenoids here, and we have completed the solution of question number 10. So now moving on to the question number 11, this question is asking us, give a brief account of viruses with respect to their structure and nature of genetic material, also name 4 common viral diseases. So in this question we have to basically tell about the structure of viruses. Also we have to talk about the type of genetic material present in different viruses, and the last but not least, we have to give 4 common viral diseases name. So if we talk about viruses, so viruses are basically the submicroscopic infectious agents. Viruses are submicroscopic infectious agents which can infect all the living organisms present on earth. The entire infectious particle of a virus is actually known as virion, and they can have genetic material. This actually the virus, if we talk about the structure of a virus, then if we talk about the structure of a virus, then they have only nucleic acids which is enclosed by a proteinaceous coat, and this coat is known as capsid. So what is actually the structure and the description of structure of viruses? Basically these are the organisms which are very complex in nature and very simple in structure. They are submicroscopic infectious agents, and these infectious agents can cause infections in all of the organisms, including plants, bacteria, in fact, and human beings, animals. All the organisms can be affected or infected by these small particles known as viruses. They are microscopic infectious agents. Now, the entire infectious material or particle is known as virion and have nucleic acids as their genetic material, which is enclosed by a proteinaceous coat known as capsid. This capsid or a proteinaceous coat is actually composed of some small subunits known as capsomeres. These capsomeres are actually arranged in a helical manner or polyhedral geometric form. So this is about the structure of viruses. If we talk about the genetic material, they can have single-stranded or double-stranded genetic material or nucleic acids in them. These nucleic acids can be single-stranded, can be double-stranded, and these actually, if we are talking about the nucleic acids in the viruses, similarly to plants and animals, they also can either have RNA or can have DNA. So the nucleic acids can be DNA or RNA in them, which can be single-stranded or double-stranded depending upon the type of viruses. If we talk about the viruses which affect plants or infects plant, then they have single-stranded RNA as their genetic material. The viruses which can infect plants, they have their genetic material as single-stranded RNA. And if we talk about the viruses, the type of viruses which can infect animals, they can have single-stranded RNA, double-stranded RNA or double-stranded DNA as their genetic material. The last but not least, the viruses which can infect bacteria. As I have already mentioned that these viruses, all the viruses can infect all the organisms present on earth such as plants, animals, humans, even they can infect bacteria, and the viruses which can infect bacteria have a special names such as bacteriophage. Mostly the bacteriophages, which are the viruses infecting bacteria, are having only a double-stranded DNA. Most of the bacteriophages, most of the viruses which can infect bacteria have double-stranded DNA. So all this is about the genetic material present in viruses and the structure of viruses. The four common viral diseases we have is AIDS, smallpox, mumps and influenza. So all these are the four common diseases which can be caused by viruses. So answer of this question goes like this. First we are going to talk about the structure of viruses. So they are submicroscopic in nature. They are submicroscopic infectious agents. They are submicroscopic infectious agents and can infect all the living organisms, not non-living objects. They can only infect living organisms. They can infect all the living organisms including plants, animals, humans and as well as bacteria. The entire infectious particle or virus particle, the entire infectious virus particle is known as virion, V-I-R-I-O-N, virion, and have nucleic acids which is enclosed within a proteinaceous coat, known as capsid, within a proteinaceous coat known as capsid. This capsid is composed of small subunits known as capsomeres. So this capsid is basically the name of a proteinaceous layer which encloses the genetic material or nucleic acids of virus. Viruses are not having cell membrane, cell wall. They are just the organisms with only nucleic acids. It can be RNA, it can be DNA. So they are the organisms which are only having nucleic acids such as RNA or DNA, which is enclosed by a proteinaceous membrane or a coat which is known as capsid. So capsid is the name for that coat which is proteinaceous in nature and enclosing the genetic material or nucleic acids in them. This capsid is basically, or the protein code of viruses is made up of capsomeres. Capsomeres, which are small subunits and are arranged. These capsomeres are arranged in helical manner. These capsomeres, which are the small subunits and composing the protein coat in viruses known as capsid, are arranged in helical manner. Or in polyhedral geometric form. So this is all about the structure of a virus. If we talk about the genetic material in them, genetic material can be different in different types of viruses. So if we talk about the genetic material or nucleic acid, it can be single stranded, or it can be double stranded. Depending upon the type of viruses. And as nucleic acid, they can either have RNA or can have DNA. So DNA and RNA are the two nucleic acids which can be genetic material in the viruses too, similarly to us. So they can have RNA or DNA as their genetic material, which can be single stranded or double stranded depending upon the type of virus. If we talk about the viruses which can infect plants, the viruses which can infect plant will have generally single stranded RNA. The viruses which infect animals, are having either they can have single stranded RNA. They also can have double stranded RNA, or also they can have double stranded DNA. So the viruses infecting animals can have single stranded RNA, they can have double stranded RNA, they also can have double stranded DNA as their genetic material. The last but not least, if we talk about the last type of viruses that are known as bacteriophages, then we know that the bacteriophage name is given to those viruses which are capable of infecting bacteria, because we know that these are the infectious materials and can infect all the living organisms present on earth. They can infect plants, they can infect animals, they can infect bacteria. So the type of viruses which can infect bacteria are known as bacteriophages, which are viruses infecting bacteria. So if we talk about the type of genetic material present in the bacteriophages, they are mostly having double stranded DNA. So the bacteriophages mostly having the genetic material or nucleic acids as double-stranded DNA. Now we have completed the structure of viruses as well as the type of genetic material present in the different types of viruses. Here we comes with the last part of this question, that is name of some common viral diseases. So if we talk about the common viral diseases, they can be AIDS, they can be smallpox, it can be mumps, and it can be influenza. Even the common cold is also caused by virus which is known as rhinovirus. So we have a huge variety of diseases which can be caused by different types of viruses, and here we have all these as example of the viral diseases which are common to known, and these are caused by the viruses. So now with this we have completed the question number 11. Now here we comes with the last question of this chapter of biological classification, and we have question number 12 on the screen, that is organize a discussion in your class on the topic, are viruses living or non-living? So we have to basically discuss that viruses are living or non-living matters. So basically we know that viruses are microscopic infectious agents which can infect all the living organisms including human beings, animals, plants, even bacteria. Now if we talk about the characters which can differentiate between the viruses, whether they are living or they are non-living. So if we want to discuss the characters of virus, that we know that the viruses are actually microscopic and they are made up of, they are only having nucleic acids such as RNA or DNA as their genetic material, which is enclosed in a proteinaceous coat known as capsid. The presence of these nucleic acids such as RNA or DNA, which can be single-stranded or double-stranded depending upon the type of viruses, the presence of these nucleic acids, RNA or DNA, suggests that viruses are alive, viruses are living in nature. Also, they respond to their environment in a limited manner when they are present inside the host cell or any host organism. So these properties, that they can respond to their environment when they are present in the host cell or host organism. Also they are having nucleic acids such as RNA and DNA. These properties of viruses are suggesting that viruses are alive, viruses are living creatures. However, some characters such as the inability to reproduce without using the machinery of a host cell or organism, and also their acellular nature, because we know that viruses are just a particle with only nucleic acid enclosed by a proteinaceous coat known as capsid. They are not having cell wall, cell membrane, they are not composed of any cell. They are just nucleic acids with a protein coat known as capsid. So their acellular nature and their inability to reproduce without using machinery of a host organism, these properties suggest that viruses are not living creatures, they are non-living objects or non-living particles. So therefore, classifying viruses has remained a mystery for modern systematics, and this is why we cannot clearly mention or we cannot classify or clearly say that viruses are living creatures or they are non-living things or non-living objects. They are non-living objects or non-living particles when they are present in the environment, into the atmosphere. Once they go inside the host organism, they are actually considered as living creatures because they respond to the environment. They respond to the environment present inside the host organism. Also they reproduces with the with actually taking the machinery of a host organism, and that is why we cannot clearly mention that they are actually living creatures or non-living creatures. So if we want to discuss then, viruses are microscopic. We already know that these are microscopic, infectious agents which can cause infections in all the living organisms, including bacteria, human beings, animals and plants. They are having nucleic acids such as RNA or DNA, and this RNA or DNA can be single-stranded or double-stranded in them. So viruses are microscopic creatures, microscopic particles, infectious agents which can cause infections in all the living organisms present on earth. They are having nucleic acids such as RNA and DNA, which can be single-stranded or double-stranded, and because of the presence of these nucleic acids such as RNA and DNA, they are considered as living creature. Also another character such as responding towards the environment when they are present inside the host organism, which also makes the property of living organism. So this property of responding to the environment also suggests that they are living creatures. So they can respond to the environment, which makes us believe to that fact that viruses are actually living creatures. However, some characters such as inability to reproduce without using the machinery of host cell, that means they cannot reproduce by their own machinery of host cell. Also the acellular nature of viruses, suggests that they are not living creatures, they are non-living organisms, non-living objects, because they do not have cell. Also, they are not capable of reproducing by their own. They need the machinery of host organism to reproduce. So all these are the properties which can tell us or which can make us believe that the viruses are not living creatures, they are non-living things. And the properties mentioned on the top of the screen, that is, they are having nucleic acids such as RNA or DNA. Also, they can respond to their environment. These properties can suggest or make us believe that viruses are living creatures. So these are the two properties which lie in between the fact that whether the viruses are living creatures or they are actually non-living objects. And because of this, therefore, classifying viruses has remained a mystery for modern systematics. Now this is the complete solution of question number 12, and with this we have completed the whole NCERT discussion of chapter number 2, that is biological classification, from class 11th. This is all for today, and thank you and have a nice day.